Stepping outside subjects the human body to a continuous bombardment of invisible, energetic solar waves. The skin forms the body's first planetary surface shield against this ultraviolet radiation. Across this surface lives a dense, teeming ecosystem of bacteria and fungi. As solar radiation strikes and penetrates the outer layers of the skin, it risks causing severe cellular damage and disrupting this delicate biological community. Surviving daylight requires an active, ongoing biological operation to manage the chemical fallout generated on the surface of our bodies. When ultraviolet radiation hits the skin, it triggers an immediate chemical reaction. A molecule normally responsible for maintaining skin moisture absorbs the energy and transforms into a compound called cis-urocanic acid, or cis-UCA. In tiny amounts, cis-UCA serves a useful purpose by preventing excessive inflammation, but as it accumulates under the sun, it aggressively suppresses local immune activity. This localized immune blackout leaves the physical barrier highly vulnerable to cellular stress, irritation, and opportunistic infections. If sunlight automatically generates a chemical that shuts down our immune barrier, how does the skin survive a single afternoon without collapsing? Without a dedicated, real-time cleanup crew to remove this immunosuppressive chemical, human skin would be incompatible with natural daylight. Beneficial surface microbes, particularly a species called Staphylococcus epidermidis, continuously monitor and respond to chemical changes on the skin. These specific bacteria contain an enzyme that allows them to consume the toxic buildup of cis-UCA. They burn it for energy, clearing the immunosuppressive molecules and keeping the skin's defense systems active. Other microbes provide a secondary layer of reinforcement. They take an everyday amino acid found on the skin, called tryptophan, and convert it into specialized defensive signaling molecules. These microbial signals lock into a skin cell sensor known as the aryl hydrocarbon receptor, or AHR, turning on emergency repair pathways. Skin cells then fuse tightly together, measured as increased transepithelial electrical resistance, eliminating physical gaps to form a stronger wall. Skin microbes operate as biological engineers, actively sensing environmental damage and rebuilding our physical armor in real time. Every solar signal hitting the skin surface immediately plunges deep into the human circulatory system. This initiates activity along the gut-skin axis, a massive two-way communication highway directly linking the exterior skin barrier to the interior gastrointestinal tract. UV exposure synthesizes active vitamin D. Skin bacteria produce AHR-activating compounds, and both messengers enter the bloodstream. They travel rapidly through the circulatory network until they reach the deep intestine, binding to receptors along the gastrointestinal lining. A localized dose of sunlight on the arm or face triggers a systemic, whole-body alert. The exterior boundary and the internal digestive system operate as one continuous organism. When the gut receives these solar messengers, it reacts immediately by tightening the intestinal walls. This optimizes the internal environment and reduces baseline inflammation. Simultaneously, gut bacteria ferment plant fibers to produce potent short-chain fatty acids, or SCFAs. These SCFAs reverse route, traveling up the circulatory network back to the skin. Upon arriving, they cool local inflammation and provide the metabolic fuel skin cells require to recover. The gut functions as a critical internal supply line, providing the specific raw materials the skin needs to maintain its resilience and repair its barrier. When poor diet, environmental factors, or severe stress compromises the delicate gut ecosystem, this vital biological loop completely breaks down. This state of imbalance is known as dysbiosis. Harmful gut bacteria overpower the beneficial species, effectively halting the production of protective short-chain fatty acids. Without constant regulation from those beneficial microbes, the intestinal wall weakens. Toxic, inflammatory bacterial byproducts slip through the gaps and leak directly into the bloodstream. Instead of receiving vital repair signals, the skin is suddenly bombarded with internal inflammatory waste. This constant irritation leads directly to chronic barrier instability, manifesting in conditions like atopic dermatitis. A weak, easily irritated, and sun-damaged exterior is often the final symptom of a collapsed internal supply chain. Protecting our biological defense network requires a specific approach. We must augment the system without disrupting its natural communication lines. Correctly formulated mineral sunscreens achieve exactly this. Physical ingredients like zinc oxide act as an external radiation filter, bouncing ultraviolet rays away from the body. Because these minerals sit strictly on top of the tissue, the beneficial microbes beneath them are undisturbed. They can safely continue consuming excess cis-UK and sending their vital repair signals inward to the gut. Maintaining this systemic relay requires both the external mineral barrier and a consistent supply of dietary fiber to fuel the internal microbiome. True biological resilience against the elements requires maintaining both sides of the barrier. We survive the environment by protecting and feeding the interconnected ecosystems living on us and within us.